into TYBSc Zoology Semester 1, paper number 2, Mammalian Histology course. We are discussing 3.4 elementary canal histology. In that earlier 3 lectures, we have seen basic gross histology of digestive system, thereafter histology of esophagus, then stomach, and we are in the middle of a discussion of a small intestine. So in earlier lecture we have seen the basic structures of intestine anatomy and basic histology structures. Now we are discussing here a cross section of a duodenum, highlighting the Paneth cells, Brunner's glands, and mucosa. So here the surface area of mucous membrane of small intestine is extensive. This is achieved by virtue of a considerable length of intestine, the presence of numerous circular folds in the mucosa, presence of numerous finger-like processes, or you can say the villi, that protect from the surface of the mucosa into the lumen, and the presence of numerous depressions or crypts or cavities that invade the lamina propria. So the presence of microvilli on the luminal surface of cells lining the mucosa. Now, if you remember the structures in a stomach or a structure in a that gastric gland, similarly here we have depressions or hollow spaces on a surface. There it is called as a crypts. Opening of a crypts or the crypts are depressions in a lamina propria. Now, how these villi are formed here? The villi are formed because the circular folds are also called as the valves of Purkinje. Each fold is made up of all layers of the mucosa lining. The submucosa also extend into the fold. Now here if you trace any single, now this is a single villi. Now what you see here is a lamina propria is also extended in that villi. So you can say it is a small tubercular growth of a mucosal layer. The folds are large and readily seen with the naked eyes. You can see here that folds. They are absent in a first 1 or 2 inches of a duodenum, immediately after the stomach, where the junction is. If you see in a first 1 or 2 inches, that large folds or villi are not there. In the whole of a duodenum, you will find these villi present. The folds gradually become fewer and less marked in the ileum, and the terminal part of the ileum have no such folds. So the highest number of villi you will find in a duodenum. It goes on reducing in duodenum and nearly it vanishes in a ileum portion. Apart from adding considerably to the surface area of the mucous membrane, circular folds tend to slow down the passages of content through the small intestine, thus facilitating the absorption. What does exactly villi do? The villi are typically a finger-like projections. These villi are a finger-like projections consisting of a core of reticular tissue covered by the surface epithelium. Here you can see all that villi are covered with the epithelial layer. The connective tissue cores contain numerous blood capillaries forming the plexus. In previous diagram we have seen that blood vessel plexus. Blood vessel plexus consist of blood, that veins and artery and the lymph tubes. The endothelial lining, the capillary, is fenestrated, that allow that rapid absorption of nutrient from the epithelial surface into the blood. Each villus contains a central lymphatic vessels. Here you will see that if you remember some earlier photograph here, that is green color portions tube is shown in that diagram. That green color tube in a these flexus or a group of capillaries present in a villi that is called as a lacteal. Distally, the lacteal ends blindly near the tip of the villus. The proximally, it ends in a plexus of lymphatic vessels present in the lamina propria. Occasionally, the lacteal may be double. Some muscle fibers derived from the muscularis mucosa extend into the villus core. Sometimes these muscles also extend into these villi portions. In some situations, the villi, instead of being finger-like, they are flattened leaf-like structures. Okay, like this. Here they are very much flattened, but still we call it as a villi. The villi are greatest and most numerous in the duodenum. They progressively decrease in size and in number in proceeding caudally along the small intestine. It has been estimated that the presence of villi increases the surface area of epithelium lining of the small intestine about 8 times. These cells lining the villi are described as: first portion is a crypt, then epithelial lining, then the types of cells, the crypt of lubricant. Now this portion here, it forms a cavity at the base of two villi. Now this cavity is called as a crypt, and here it is named with the crypt of lubricants. They are tubular invaginations of epithelial into the lamina propria. They are really simple tubular intestinal gland. We can compare them with the gastric glands that are lined by the epithelium. Now that epithelium is supported on the outside by a basement membrane, the cell lining the crypts are considered separately as Paneth cells, mucoid cells. So what exactly that epithelial cell lining is? The epithelial covering the villi and areas of the mucosal surface intervening between them consists predominantly of columnar cells that are specialized for absorption. These are called as enterocytes. They scattered the columnar cells. There are mucus secreting goblet cells. All these white vacuoles you see here, mucus secreting goblet cells. The cells lining the crypts here internally are this portion. The cell lining the crypt are predominantly undifferentiated. These cells multiply to give rise to absorptive columnar cells and to goblet cells. Near the base of the crypts there are Paneth cells that secrete enzymes. These pinkish color structures are the Paneth cells which produce enzymes. Endocrine cells bearing membrane-bound granules filled with various neuroactive peptides are also present there. The various cells mentioned see in detail as a individual cell type. So we'll continue with the Paneth. These Paneth cells are found only in the deeper part of intestinal crypts. They contain prominent eosinophilic secretory granules. Paneth cells are seen to contain considerably rough endoplasmic reticulum. If you see under electron microscope, you will find that they contain lot of endoplasmic reticulums. Other organelles that and some irregular microvilli are present in that Paneth cells. These cells are rich in zincs. The function of zymogen cells or Paneth cells is not very well known, but they are known to produce lysozymes that destroy bacteria. So overall, these cells, they also produce enzymes, and basically these cells are role in a defensive mechanisms. Now in this photograph also we can see here Brunner's glands embedded in a submucosa portion here, Brunner's glands. These are the compound tubulo-alveolar glands. Their ducts passes through the muscularis mucosa to open into the crypt of lubricant here. They have ducts that open into this crypt of lubricant. The cells lining the alveoli of duodenal glands are predominantly mucus-secreting columnar cells having flattened basal nuclei. When these mucoid-producing cells are found less to produce mucus, they are supported with these Brunner's glands. Brunner's glands are basically a mucus-producing duodenal glands. The duodenal glands are most numerous in a proximal part of duodenum. They are few or even vanish in a distal part. The secretion of the duodenal glands contain mucus, bicarbonate ions, and an enzyme that activate trypsinogens that produced by the pancreas. That is the significance of these Brunner's glands. They have secretions not only of a mucus but also a bicarbonate ions to neutralize gastric acid entering into the duodenum, and production of trypsinogens, the enzymes that activate trypsinogens coming from the pancreas. The proximal part of the here jejunum, they are less. So in this photograph we are seeing the different mucus secreting glands, epithelial lining in the duodenum, and the Paneth cells present in the crypt of lubricants, and the opening of a Brunner's glands into this crypt of lubricant. Now in case of jejunum, the proximal part of jejunum shows a significant difference in structure from the terminal part of the ileum. Okay, and the duodenum, this portion is very much different. They have the difference is based on the features, like the large diameter size of this villi are very large, thick wall, and large and more numerous circular folds are seen here. Okay, here crypt is very much reduced and villi portion is rounded with lot of goblet cells. In case of ileum, the pointed structures of villi is very much reduced. You see that villi are very blunt shape and large in the diameter, and with lot of increased number of goblet cells. Paneth cells are also present at the base, and in more increased number of Pairs patches are seen in a ileum. Now, in this color photograph, you can see macrovilli, goblet cells, Paneth cells, crypt of lubricant. But here you can see a large bulging structures of some different pattern. So here, in addition to the scattered lymphocyte, there are present in a lamina propria large number of solitary follicles or isolated lymphatic nodules, more numerous distally in the intestine. If large, they may occupy the entire thickness of the mucosa and bulge the surface. There are no villi and may be no crypt on the surface of large follicles, which are then separated from the lumen only by a simple columnar epithelium. Okay, in many regions, but mainly in the ileum, follicles may be so numerous and close together as to aggregate into a large mass of lymphoid tissue visible to the naked eyes. They vary in size from around 12 to 20 millimeter long and 8 to 12 wide. The large axis lying along the length of the intestine. Always they are situated on the anti-mesenteric borders, that is on the side away from the attachment of the mesenteries. These are called as a Pairs patches or aggregated nodules of lymph. From the immunological point of view, the lamina propria is important, with lymphoid cells and macrophages providing a barrier between the body and the antigen, microorganisms, and other foreign materials that always are present in the intestinal lumen. This is a very they have very much role in a defensive system in our small intestine. Now here in a large view you can see a presence of this circular ileum microvilli and this large number of goblet cells here on the surface, and you can see the change structures of the microvilli here. Small intestine penis cells are shown here in a large view with the accumulation of these secretory granules on the surface. So nucleus located at the base, and centrally they are pouring their secretions here. These are the penis cells. Here again another photograph of a small intestine. You can see these pinkish color structures as a Paneth cells secretions in this lumen. This is a crypt or a hollow space is a crypt of that lubricant coming from the villi. And then these are here in this submucosal layer. You will also find some flexes, a stretch response flexes, Meissner's flexes. You can see here. This is a mucosal layer, and these are the Paneth cells in the crypt of liver. Now we start with the discussion of this large intestine. So major site of absorption of water and salts take place in a large intestine. Mucosa, with respect to mucosa, if we compare it with the other intestines, so no villi are there. Glands of lubricant with many goblet cells and no Paneth cells. There are the glands, but here that glands contain more goblet cells and no presence of a Paneth cells. In submucosa, there are no glands embedded, which are there in a duodenal glands or gastric glands, the esophageal glands. Same way here in large intestine, in submucosa, there are no such glands are present. Then muscularis externa, that is inner circular muscles plus outer longitudinal muscles with the mesenteric plexus between these two layers. Outer longitudinal muscles create three narrow bands called as a tinea coli. Four anatomic sections you can see here: the circum, appendix, colon, rectum, and anal canal. So you can separate it into the this ascending, transverse, descending uh, arm of a large intestine. This is subdivided into cecum, appendix, colon, rectum, and anal canal. The sac-like cecum is there that lies below the ileoceliac walls in the right iliac fossa, containing a worm-like vermiform appendix. So, visually, you can mark the presence start of this large intestine with the presence of this appendix. So that has a distinct regions of colon, ascending colon, then hepatic plexus, that is the turn portions, then a transfer colon, then the turn portion, that is sphenic plexus, then descending colon, and the turn portion, that is sigmoid colon. This is all about the anatomical position of a large intestine. So here, from here you can start with this ileum portion. Ileoceliac wall is there, and from here it start the large intestine. So start with the cecum, and cecum, this for appendix is attached to the cecums. Then these portions is a transfer. This portion is a ascending colon portion, and transfer colon. This plexus, turning plexus are there. Descending colon is there. Sigmoid colon and the rectum. So colon, transverse, and sigmoid portions are anchored via mesenteries called as a mesocolon. The sigmoid colon joins the rectum, the anal canal, the last segment of the large intestine open to the exterior at the anus. We don't go in much detail of these anatomical structures, but we are more focused and interested in a histological part of this large intestine. So if you see the mucosa of large intestines, do you see any villi or microvilli on a surface of inner surface of this mucosa? No. So here vanish of a villi take place. No villi you will see here. But what else you see? simple cuboidal columnar epithelial, and majority of that columnar epithelials are converted into the goblet cells, secretory cells as a goblet cells. Then also some goblet cells are grouped together and forms a major mucus producing glands which has a ducts to open outsides, and so large intestine mucosa is embedded with large number of goblet cells. Here in colon portions, this mucosa portion is there, submucosa portion, and the muscularis externa circular portion is there, which is full of muscles. Okay, in actual photograph, you can see this structure here as a that mucosal layer. Muscularis externa, you can see the inner circular muscles, outer longitudinal muscles, then mesothelium and the serosa portion. Here the submucosa, and above there is a mucosa portions. All these submucosa portions is full of all these mucosal glands are there. Now appendix is non-functional in a human, but it may be functional in other mammals. So the appendix, there you will find large number of lymphatic nodules and the same structures like large intestines with some amount of villi and crypt and secretions of mucus. In anus, it is a internal sphincter portion is there with the smooth muscles and external sphincters with the skeletal muscles. So anus rectal junction is shown here. So this portion is all about the large intestinal portions with lot of mucus producing goblet cells, mucous glands are there, which are vanish in terms of this um, anal portion where there is a only a epithelial layer is also changed into the stratified squamous epithelial type. So if we start to summarize the histology of digestive system, we can summarize it in this way. The organ is given on one side and the type of epithelium, how it goes on changing from the organ to one organ to another. So in esophagus, you will find that it is a stratified squamous epithelium. In stomach, it is a simple columnar. In a small intestine, also simple columnar. In large intestine, also simple columnar. Only in anus portion, it is stratified squamous epithelium, same as the esophagus. Then the fold of epithelium, if we want to check how folding occurs in epithelium, in case of esophagus, there is no folding. It is a plane. In stomach, it forms a huge folds, and that is called as a rugae, and small fold as a called as a gastric pits. In case of small intestines, these large foldings are called as a plica circulari or a villi, and that forms further that foldings forms a crypt of lubricants, and again over the epithelial cell you will find there is a microvilli. So folding is highly increased in a small intestines, whereas in a large intestine there is a large fold or hostra is there, and a small fold is a minimum fold is of intestinal glands which produce that mucus. Then if you want to see the what specialized structures are present in different organ in the digestive system, so in esophagus you will find there is a submucosal mucous glands are present. In stomach there is a no specialized structures as such. In the duodenum there is a Brunner's glands, or that again the mucus producing duodenal glands are there. In ileum, we'll see the large lymphoid nodules called as a pear patches. In large intestine, no such modification is there. If we compare the smooth muscles layers among the different organs, so in esophagus, we'll find there is a circular and longitudinal muscles are there. In stomach, oblique, circular and longitudinal type of muscles are present on outer layer of this wall of digestive systems. In small intestine, again the circular and longitudinal, and large intestine, again the circular and longitudinal. So these esophagus, small intestine and large intestines, they have this only two types of muscles that leads to the formation of peristaltic movement or propulsion of food forward. Whereas stomach have one more oblique kind of muscles to churns or to mix the food or to maintain the elasticity in stomach. You will find one more kind of a muscles is added. Okay, if you compare the serosa of different digestive system organs, what you see here is the esophagus. That serosa is called as adventitia due to the fact that their esophagus is not in a cavity, so it is covered more with the connective tissue, and serosa instead of serosa it is called as adventitia. Here in stomach, it's a visceral peritoneum forms the outer layer. Again, in small intestine, large intestine, the same visceral peritoneum form the outer layers. But when it reaches to the anus portion, there that outer portion is covered with the adventitia and not the serosa.